All right. So the thing about an action potential is there's going to be a refractory period. There's going to be a refractory period during which no matter how much stimulus you get, you're not going to be able to initiate a new action potential. All right? So since it's all or none, oops. Here we are. Here's resting membrane potential. And graded potentials begin to change the local currents from graded potentials begin to impinge upon the axon hillock, right? Because a lot of graded potentials around the entire soma cell body have converged on the axon hillock, and we begin to slowly depolarize until we reach this threshold. It's only 10 millivolts different. At minus 60, that sodium channel, the voltage-gated sodium channel swings open. Boom! Influx of sodium, rapid depolarization. Potassium channel opens, sodium channel closes, and we get repolarization. So as soon as we reach threshold, once you've reached threshold, we are going through this process. You can't initiate a new process until you've finished it. It's like jumping rope. Okay? If I'm going to jump, I can't initiate a new jump until I've landed from the previous jump. Okay? So, whereas with the graded potential, I can dump some chemical on, depolarize. I can just dump some more chemical on and depolarize further. Have a more intense stimulus in the area. I mean, you can saturate the chemically gated channels, yes.
So I guess the way to describe it would be instantaneous membrane potential right at the point where the graded potential is happening. Do those reach the same, what's it called, the same amplitude as the action potential, or are those always smaller than an action potential?
A graded potential?
A graded potential, like right at the maximum spot of the graded potential. Like how
the
big
depolarization is going to be based upon how many channels are open. If you have only a single channel open, there's only so much of an impact on the membrane potential that a single channel is going to be able to have. And so that would be a modest bump. You know, you're not going to be able to have a full depolarization to plus 30 before the acetylcholine or whatever the neurotransmitter is going to diffuse off of that channel, and the channel is going to close. All right, so there is a mass effect depending upon how much neurotransmitter is dumped on that membrane.
But say you have like a synapse, you have like a moderate amount of receptors.
Mm-hmm.
Are you going to get at that one spot? Is it going to reach like negative 30?
No. So at a synapse, and we'll talk about synapses, but at a synapse, there is going to be a. So if it's a neuromuscular junction, it depends on what kind of a synapse it is. But at a neuromuscular junction, there's a synapse. Those chemically gated channels are right next to voltage gated channels. So those chemically gated channels only need to reach threshold. They don't need to reach plus 30. In terms of a dendritic, a synaptic dendritic contact, that is going to be an amplitude modulated function. So it does not have to reach plus 30 right in the site of stimulus. Okay, so you can imagine like the extreme case of a single receptor, a single channel that is going to give you some sort of bump in the membrane potential, and then the more channels you add to that, the greater that is going to be, right? The greater the depolarization. But the key here with graded potential, it's not just about the membrane potential. The membrane potential is a reflection of the local currents of sodium. It's those local currents which are traveling away from the site of the stimulus, which are going to enable excitable membrane to reach threshold. You following that one?
Yeah, I'm following. I get how it works.
Maybe I'm not understanding your question.
Well, I think you basically answered my question already. So let's just
Okay.
go on and talk to you about it after.
Okay. So there's this absolute refractory period during which absolutely you cannot start a new action potential. However, there is a point at which we've repolarized. It is hard to start a new action potential. Like, you have to have a lot of local current, a lot of graded potentials impinging upon that excitable membrane to initiate a new one. But it is possible. It is possible. Not normal stimulus. It has to be a werewolf has just entered the room and is ready for lunch for you to get a strong response in this absolute refractory period. All right? So then all the channels are closed, and we need to reset the relative ratios, the relative electrochemical gradients of the cations involved. All right. So once we have started this at the axon hillock, this depolarization is going to propagate. And this is the temporal profile that I just showed, at a single point in the membrane, how the membrane potential is changing with time, right? But that translates into a spatial profile as well, where along the axon, this depolarization is traveling in a wave that goes along the length of that axon. And that's what this is meant to convey. I have a nicer diagram of that right here. So we've stimulated something at the axon hillock where there was depolarization. This is the absolute refractory period, where you can see we flipped the membrane potential, and it's traveling this way. It can't travel backwards. It can't travel backwards because of the refractory period. There's this wave of refractory period behind it that's causing it to travel down the excitable membrane that hasn't yet experienced this depolarization, this action potential. Okay? And it propagates this way. Does that make sense? Does anyone not understand the dynamic of that? Okay. So there's two methods. This is a concept that I could have left to maybe Dr. Martin's class, but I'll just touch upon it. There's the notion of continuous propagation and saltatory propagation. So continuous propagation is what happens in unmyelinated axons, which is a tiny minority of the axons in your body. A very, very, very tiny minority of the axons in your body are unmyelinated. But unmyelinated axons are in squids. Giant squids have these giant unmyelinated axons, and a lot of work has been done in unmyelinated axons of the squid because it's a little bit simpler case. The electrophysiology of it is a little bit more amenable to experimentation. Anyways, this is the way this works is the way you think it would work. We have an action potential that initiates here. Local currents travel away from the point of depolarization of these electrically gated channels. We begin to reach threshold here. So we reach threshold at the second sort of segment. This is a continuous thing. We're breaking it up into segments, but this is a continuum of electrically gated receptors along the cell surface. So this is going to reach threshold, and now these sodium channels are going to open and continue to have this spreading wave of local current as it travels along the length. So here we go, etc. All right. Unidirectional, and this continuous propagation, it says here about 1 meter per second, which is actually not super fast. That's not exceptionally fast. It seems like it might be, but it's not. Okay, the second type is this saltatory propagation. And So in a wire, like the wires up on the telephone poles, like the electrical power grid, when that was first envisioned by Nikola Tesla, the first wires that they began to string up were uninsulated. They were uninsulated. And of course this was because it's easier to make uninsulated wire. This is, of course, dangerous for a number of reasons, but more than that, it was inefficient. Who here has taken physics, the electricity and magnetism portion of physics, right? So you will remember from that when you have a charge carrying medium like a wire, and you put so as current passes through that charge carrying medium, there is an electrical field that is generated perpendicular to the flow of electricity. All right, anybody who's taken a nail and wrapped a wire around it and stuck a battery to it can see that there's an electromagnetic field that's generated perpendicular to as the nail becomes magnetized. So this is energy that is in the electricity that's flowing being transferred into the generation of an electric field. However, if you insulate it, if you insulate the wire, it is containing that electrical field, the energy of that electrical field to the medium, the charge-carrying medium. So if you insulate an axon, if you put layers of myelin, which is just these fatty membrane layers, these lipid bilayers, stacks of them, around a wire, you are containing the energy from that electromagnetic field that's being generated by charge carriers, by this local current. You're concentrating that in the axoplasm here, all right? So what happens is we call these areas where there's myelin internodes. However, you've seen on the phone wires, the telephone poles, there's these big trash cans every so often, right? Transformers. Those are stepping the power back up. They're stepping the power up. And that's essentially what these nodes are. So here is the myelin. We call this the internode. And then there are these intermittent nodes, these nodes every so often. And what happens is we have a depolarization event at the membrane here, and the current jumps quickly because of this insulation to the next node. However, there's a power drop. There's a power drop associated with that. And the distribution of these nodes, once it gets to this point, the length of this, how am I saying this the right way? The length of this myelin internode is such that the power drop is just enough so that when this current gets to the next node, it's enough to bring this to threshold, all right? And this has another depolarization event that happens just on this membrane here, which jumps to the next node, depolarizes here, jumps to the next node very rapidly. Yes, Jacob?
What is the connection? Like, what's happening in that section of myelinated axon to communicate that current? Is it just traveling through the cytoplasm of so it's just the sodium ions traveling through the cytoplasm?
Yeah, there is local current that is happening, but the efficiency of that process, the power that is being put into the system is being put into it more efficiently. Less of it is being dissipated as an electrical field that is perpendicular to the direction of electrical current. That's as plainly as I can state it for you. It's containing the electric field. So the electric field power that would be going into a perpendicular electric field is not being dissipated into that perpendicular electrical field because it's being contained by the myelin. And so it's a more efficient way of sending an electrical current along the parallel axis of transmittance. still are giving me a skeptical look.
Yeah, I'm not entirely sure that that's the exact way that insulation on the wire works.
Okay.
But anyway, I think for the purpose of this.
Okay, well,
But yeah,
enlighten.
I don't remember exactly. I don't want to get into it. It's not a physics class.
Okay. Well, the nuances of the physics aside, we have depolarization at a membrane, local current, and this local current rapidly jumps to the next node, depolarization, and so forth on through this chain. It happens much more rapidly than continuous propagation, and it requires a much smaller diameter of the axon. Continuous propagation, V equals IR, that R term, the resistance to current flow, is directly proportional, or inversely proportional, pardon me, to the diameter of the wire, if you will, or the axon, the exoplasm that the current is passing through. So we'll see that in just a moment here. And this is just a cartoon of that. So yeah, here is the relationship of the speed of propagation and the axon diameter. So the speed of the propagator, the velocity, the directional speed of propagation is proportional to the inverse of the capacitance. So how much energy is how much electrical field energy is that axoplasm able to store? All right, and then we have the axonal diameter is directly proportional to that, and you can imagine that it would be inversely proportional to the capacitance. And then we have membrane resistance and axonal resistance. So the membrane resistance is what we're increasing when we increase the insulation, the myelin. By increasing the myelination, we're increasing this term right here. Axonal resistance is obviously inversely related to the axonal diameter. Okay, so, and this is the relative scale that we're talking about. We can have scales that are less than 1/10 of a meter per second. So only about that far within a single second. That's going to be continuous propagation, very slow. Whereas over here, up to 100 meters per second, 580 miles per hour. That's quite rapid. That's quite rapid, depending upon the level of a number of factors, but primarily axonal diameter and myelination of that axon. All right, you guys have questions on this so far? Oh yeah, so the point I was making is you have to quadruple the diameter to double the velocity, because this is a root. And then myelination is a great way to reduce this term as well. So the membrane capacitance, when you insulate it, you're reducing the capacitance of that axon, because you are sequestering that the electrical field is not getting projected perpendicular to the axon. It's getting it's being contained within the axoplasm for the propulsion of the signal. All right. So this is the last concept I wanted to hit here. The all or none principle implications. So an action potential is either going to be triggered or it's not going to be triggered, right? So this was the all or none. We either reach threshold and have an action potential, or the graded potential does not quite reach threshold. It is, as Logan pointed out, binary. An action potential is binary. Either happens or it doesn't happen. So this is sort of a digital informational transfer via the synapse. And because of that, it's frequency encoded. And here's what I was trying to draw on the board earlier. So with amplitude informational encoding, the strength of the depolarization is where we're encoding the information. All right. So we can have really strong depolarizations or small depolarizations, but the information is not necessarily encoded in the frequency. Whereas in action potential, it's how frequent are we triggering an action potential. It's all action potentials for that particular axon are going to be the same intensity. All right, because it's just a mousetrap, and if you spring the mousetrap, it's always the same strength. Okay. But the information is in how frequently you're going to trip the mousetrap. Does that make sense? Okay, are there any questions on this? I already went through this chart. Let me see, is there any point that I didn't make in the beginning of class here? Yeah, there's no refractory period integrated potential. I think we covered all these. Okay, so if there are no other questions, I'm going to jump into muscle tissue now. My favorite part of any new topic, the haiku. Sing you of sinew when you sing a new tune of when you grew anew. It's a question. Okay. So I had talked about Avicenna in the beginning of the semester. The famous Persian anatomist, physiologist from the 11th century. Here is his translated, admittedly translated quote that relates to this. The power of locomotion is that which contracts and relaxes the muscles, whereby the members and joints are moved, extended, or flexed. This power reaches the limbs by way of the nerves. And there are as many forms of power as there are of movement. Each muscle has its own peculiar purpose and obeys its decree of the composite sense. So right here in this single quote, he's pretty much summarized everything that I want to say about this unit, about muscles. So obviously muscles serve the function of motion. It's the contractile tissue in the body. And it is by action upon the levers of the skeletal system. And then importantly, we're going to spend a lot of the next several lectures talking about how this happens through the action of nerves. So nerves are going to synapse on muscle fibers and cause a series of depolarization events, which are going to lead to what's called the excitation-contraction coupling. We're going to look at that on a molecular level and understand how it is that muscles specifically contract. And then I'm not actually going to talk about the in this class. I'm not going to talk much about the skeletal system and the unique the unique functions of each individual muscle group, but that's for a later date in your education. All right, skip forward about 400 years to this guy Andreas Vesalius. I think I also have talked about him a little bit before. He's certainly beloved of the thinking medical community, the historical medical community. He was a Flemish physician who, remember I had talked about Galen and how during the time of the Romans it was forbidden to conduct dissection of human cadavers. Well, Galen got around that by being the physician to the gladiators, and he got to see all kinds of butchered humans. But he wasn't able to just dig up a corpse and cut it up or whatever. This guy here, things had changed by this point. The Renaissance was nascent, and Vesalius began to do these very careful, very meticulous dissections of human cadavers and illustrated them in exquisite detail. All right. So a lot of our discussion, not today, I'm not going to get there today, but on Wednesday is going to be focused on this, which is the neuromuscular junction. Here is a neuron. These are that's an axon. These are telodendria. This is a neuromuscular junction with the synaptic boutons on the surface, on the sarcolemma of a muscle fiber. This is where the action happens. This is where it all starts. Okay. All right. Muscle tissue. It is, of course, one of the 4 primary tissue types, and there are 3 categories of muscle tissue. Skeletal muscle is the most commonly regarded. This is muscle that is voluntary, meaning that it is under the control of the little person sitting behind the levers up in your dome. It is striated, meaning when you look at this muscle tissue under a microscope histologically, you can see stripes. It's striped, striated, and we'll talk about the functional significance of that in a moment. And these muscle fibers are multinucleate. I'm gonna ask you why that is when we a little bit down the road, so you can begin to ponder that now. What does it mean to be multinucleate, having many nuclei in the same cell? How did it get that way, and why is it that way? Functionally, what purpose does that serve? So that's skeletal muscle. The next category would be cardiac muscle. It has the similarity with skeletal muscle in that it is striated as well. So whatever functional significance this striation has, it has in common with skeletal muscle. Okay? And we'll see what that means in a bit. However, it is mononucleate, and cardiac muscle is involuntary. You do not choose to beat your heart each time your heart beats. You can affect the rate at which your heart, voluntarily affect the rate at which your heart beats, but you do not initiate contraction of your heart rate with your mind. The third category has in common with cardiac muscle that it's also involuntary. You do not initiate contraction of smooth muscle fibers. It also is mononucleate like cardiac muscle. Unlike cardiac muscle, it is non-striated. Non-striated. Okay? So, despite it having these 2 similarities with cardiac muscle, it is more dissimilar from the other 2 categories than similar, because the functional unit of the sarcomere, which is what gives rise to the striation, does not exist in the same way in a smooth muscle fiber. We will just glancingly discuss smooth muscle at some point here. The muscular system per se is only composed of skeletal muscle. All right, just skeletal muscle is the muscular system. Cardiac muscle is part of the cardiovascular system. Smooth muscle is found throughout a whole host of different organ systems. Okay, so here is a feature of muscle tissue that is going to be reminiscent of similar features in other organ systems, notably the nervous system. Okay, there are 3 layers of connective tissues in muscles, and we'll make analogies in the nervous system when we get there. The first layer of connective tissue is epimysium. It's a collagenous sleeve that surrounds an individual muscle, a single muscle, and it connects that muscle to the deep fascia, the deep connective tissue layer of the body, and it helps to distinguish a unique muscle, right? The epimysium. And so here is a muscle in cross-section. This is the collagenous sleeve that surrounds that muscle. Within a muscle, we're going to notice that there are these bundles. Each bundle is called a fascicle. A fascicle is itself surrounded by a connective tissue sleeve called perimysium. So the perimysium is the conduit for all of the VANs, the neurovascular bundles, VAN, vein, artery, nerve. So veins and arteries and nerves tend to follow similar courses through the body. They coalesce together through these sort of utility channels, I guess, is a way of thinking about it. The perimysium acts as that conduit for the muscles. So it brings nervous innervation, it brings blood supply to and from the muscle tissue through this perimysium. So all around here, investing this muscle, surrounding each of the muscle fascicles, is the perimysium. Oops. And then the third layer is the endomysium. So now if we were to look at a muscle fascicle in cross-section, it itself is made up of individual muscle cells, or we call them fibers, because that's what they are. They're not round and amorphous, they're long. Skeletal muscles can be exceptionally long. So here's a muscle fiber, and it is surrounded by this endomysium. This is where the arterioles and nerves break down into, the arteries and veins break down into arterioles and venules and then actually capillaries. So the endomysium contains a sort of capillary sleeve that is going to surround these muscle fibers. This is also where we find myosatellite cells. These are stem cells, muscle repairing, muscle creating stem cells. Anything else that I want to say there? No. Okay. So a skeletal muscle fiber, as I've just indicated, they're profoundly large. They're huge, very long, large, and are created by the fusion of multiple, often many hundreds, of mesodermally derived cells called myoblasts. These are truly muscle stem cells. Because of that fusion of myoblasts, we retain the nuclei of these myoblasts and form a long muscle fiber with nuclei distributed throughout the length. Why? Why? First of all, let's start with why is it an advantage to be so big? Way in the back. How are you? Good. What do you think? So let's start let's start with our presumption of the connection between form and function, right? And we know, we're trying to rationalize the form. We know the function. What do these cells do? What does a muscle fiber do? Contracts, right? Yeah. So, how do I put this? Okay, Logan.
So if the purpose of the cell is to have some sort of like coordinated contraction, then if you have fewer muscle cells, it'd be easier to coordinate them together because you don't have to send as many stimuli to individual cells.
Absolutely, absolutely. That's an important component right there. That's one. What else? What do you have to say?
Well, the attachment points in the skeletal system are far apart, so you need a contractile piece of machinery that can reach that long.
Okay.
And it also has to be structurally sound, because you could have a bunch of like single cell ones lined up end to end, but then that probably would not be as cohesive and not as organized.
Or unified,
Yeah,
right?
unified.
So it would be dependent upon cell-cell adhesions more than if it was just a single unit that was contracted. Okay, so now that's fine, but why don't some of those nuclei just go quiescent, get resorbed when those myoblasts fuse? Why do they retain all these nuclei distributed throughout the cytoplasm? What do you think, Matt? You're puzzling over there. You have no thoughts?
No.
What about you? You already spoke today. Let's get someone. What do you think? What does a nucleus do? What does the nucleus do?
Powerhouse has all the DNA in it.
It has the DNA, and what do we use DNA for? Making proteins. Absolutely. Okay. Why is that important in a muscle cell? Don't be afraid.
Would it constantly needs to make more muscles? So it needs
You got it. You knew it. You had it. You had it all along. A muscle cell is pretty much nothing but protein. It's pretty much nothing but actin and myosin. It's all protein. And if we need to build the muscle cell larger, if we need to repair some damaged muscle tissue, we have nuclei all over this giant cell. So if, say, we have some damage down here, we tore this muscle cell, the nuclei down here doesn't have to make the proteins and somehow ship them all the way down there. Okay? It's got a local, this is about, just like Amazon, for example, has regional distribution centers. Okay? There's not one warehouse in Taiwan that's shipping stuff all over the globe. I mean, there probably is, but it's going to regional distribution centers. Jacob, you've been patient.
Yeah. Is there a specific location, I guess, where the ribosomes are sitting? Because there's all that space being taken up by actin and myosin, or are they just synthesizing in the space that's not taken up by actin and myosin, or is there sort of localized to the edges?
Let's get into talking about the structure of the sarcomere a little bit, and we'll see Yes, it fits in amongst the sort of regular lattice work of the cell, but let's look at that in more detail. Okay. Are there any other questions there? All right. So is there any point I need to make here? No. They're long, 30 centimeters. That's pretty big. 30 centimeters is like that. A single cell in your body can be that big. That's crazy. Wow. Cool. That's pretty amazing to me. I don't know. It's just like a single cell that's that long. All right. So here's a little bit of a look. Yeah, this class is not anatomy, but we do obviously need to learn some anatomy here so we can talk about how this system works. All right, and we're going to go through it. So here's the muscle fiber, and just some terms. Sarco comes from the word sarx, and I do have this on one of my slides somewhere, maybe not, I don't know, but S-A-R-Y-X, sarx, S-A-R-Y-X, yeah. It's a Greek word that means flesh. And so sarco, the prefix sarco, simply refers to anything that means muscle, okay? So the sarcolemma is a suffix that means layer of some sort, all right? So the sarcolemma is simply the plasma membrane of a muscle cell, okay, a muscle fiber, the sarcolemma. And let's look closer now. And we see these nuclei distributed around. When we look closer, within a muscle fiber, we see these what are called myofibrils. Myofibrils. So just like, and I have a slide that makes this point in a few slides, but I'll say it now because I'm not going to get to it today, just so you can think about it. There is a fractal quality to the structure of muscle, meaning there's this scale invariant self-similarity, all right? Depending upon, come on in. My kids are here. Hi guys, just park it over there for a couple minutes. There's this scale independent self-similarity where we had a muscle fiber, a muscle that was made of fascicles, a fascicle made of muscle fibers, a muscle fiber made of myofibrils. They all kind of look the same. The organizational structure looks the same as we go up and down here, okay? So make sure you don't get confused as to which level in the hierarchy you're at. These myofibrils themselves are made of thick and thin filaments. We're going to get into that next time. Each myofibril is coated in this sleeve, here depicted in blue, which is called the sarcoplasmic reticulum. Sounds a lot like endoplasmic reticulum, in fact. It is, but it is unique to a muscle cell. So the cytoplasm in one of these cells is called sarcoplasm, all right? Whatever analogy you want to a cell, just take the prefix off and replace it by sarco, all right? So there's these cytoplasmic reticular sleeves around each of these myofibrils. What's interesting about this is they are broken into segments where at the end of each of these segments is this pouch. And this pouch is called the terminal cisterna. The terminal cisterna. A cistern is like a big well, not a well, but a big holding container for rainwater, right? So, and it's terminal to this segment here. The terminal cisterna is adjacent to what's depicted here in yellow as, we call these T-tubules or transverse tubules. These are tubules which are simply extensions of the sarcolemma down into the body of the muscle fiber. Okay? If you were a tiny little creature, you shrunk yourself down super tiny and you're walking on the surface of this muscle cell and you walked down into that hole, there's a hole there, you would suddenly be inside a T-tubule. It should not come as a surprise that the sarcolemma is excitable membrane. There's going to be voltage-gated channels on the sarcolemma, and that continues down into these T-tubules. Okay, you following me? So that this is a way of getting really distributed, coordinated events happening throughout the cytoplasm. When there's a depolarization event, getting it coordinated throughout the cytoplasm of the entire muscle cell. A T-tubule adjacent to two terminal cisterna is something we call a triad. A triad. It's like a T-tubule sandwich on terminal cisterna bread. You follow me? Pretty simple, basic anatomy here of a muscle cell. And then embedded throughout, we have these mitochondria. The cartoon does not depict the ribosomes that Jacob is interested in, but that is the basic cellular anatomy, and that's what we're going to need to pick up next time and talk about the excitation-contraction coupling, which is how all this machinery actually works. Thank you. Are there any questions? I'm pleased to have each and every one of you in my class. Yes, Logan.